This is the phylogenetic tree of chordates, the group of organisms we, as humans, belong to. And according to this diagram, humans are technically fish. Well, that is if you count two of trout, sharks, or coelacanths as fish. So how does this make any sense? You may be thinking that only an idiot would call a human, bird, or whale a fish, right? Well, hear me out. And let's see how phylogeny works. It's simpler than you think. Phylogeny is one of the many ways we classify organisms, and it's generally the most accepted method, as it classifies organisms based on their evolutionary relationships, and incorporates DNA and what organisms they are closely related to. The monophyletic group, or clade, is the basis of phylogenetic trees. A monophyletic group, or clade, contains a common ancestor and all of its descendants. So once you're in a clade, you can't evolve out of it. This is very important. A paraphyletic group, such as non-avian dinosaurs, contains a common ancestor and some of their descendants. This is not a clade, and not useful in determining phylogenetic relationships. And a polyphyletic group, such as birds that are blue, contains organisms from multiple groups, regardless of their relationships. This is also not a clade, and is useless in determining evolutionary relationships. So these top ones are clades, but these bottom ones are not. Clades can also be nested within each other, and being in a smaller clade does not exclude you from a larger clade. This is why humans can be considered apes, primates, mammals, vertebrates, and animals, all at once. Let's simplify this down. Here's a phylogeny containing the brothers Mike and Tom, as well as their cousin Greg. You can see by this node, which is the last time Mike and Tom shared a common ancestor, that Mike and Tom have a more recent ancestor than Mike, Tom, and Greg, because Mike and Tom are brothers. So this first node would be their parents. Well, Greg is their cousin, so this farther node is the last time they all shared a common ancestor, their grandparents. This means Mike and Tom are closer related, and their DNA is more similar than the DNA between Mike and Greg, as they're less closely related. And since Mike and Tom share the same common ancestor as Greg, Greg is equally related to both of them, since he's their cousin. And the relationships between the three remain the same whether the diagram looks like this, or this. We can also add John to the tree, who shares an even less recent common ancestor. Maybe this farther node is their great-grandparents. But in the end, it doesn't matter what the name of the individual or species at each node is, as phylogenetic trees focuses on the relationship between each branch. And we can make a clade from each node and all their descendants, something like this. Now the names of those clades are up to me. Say I want to make a family clade that includes Mike and Tom. I can choose to include Greg or exclude him. That's up to me. But there will always be a clade uniting any number of individuals or species that includes their common ancestor. Well, as far as we know. Maybe there's some alien bacteria that breaks this rule. So back to the fact that humans would be fish if two of trout, sharks, and coelacanths are counted as fish. Here's a simplified tree of chordates, where the relationships between groups were determined by multiple analyses of the DNA of these organisms. As you can see, tetrapods emerged from within the Sarcopterygi, also known as the lobe-finned fish, a group which contains the coelacanths, which is an aquatic organism that breathes via gills, and that most people would call a fish. The sister group to the lobe-finned fish is the Actinopterygi, also known as the ray-finned fish, which contains animals such as trout and tuna, which I'm pretty sure everyone would consider fish. Sister to the clade containing these two groups is the Elasmobranchs, which contains sharks and rays, and many people would consider sharks fish. And sister to that whole group is the jawless fish, such as hagfish and lamprey. So if I wanted to make a clade called fish, I could include just the ray-finned fish to avoid including tetrapods, but since coelacanth and sharks are generally considered fish, it should be included as well, and it makes sense to include them. And I could include the jawless fish or not. But there's no way to include sharks and trout, or trout and coelacanth, in a monophyletic group that excludes humans and the other tetrapods. And in that case, unless you think only the ray-finned fish count as fish, then you too are a fish. Well, assuming you're a human and not that alien bacteria or an invertebrate. So how do we determine where an individual or species belongs in a tree? Well, you can estimate these relationships either with morphological characteristics, as is usually done in fossils, or genetic differences. And it's done in practically the same way, by either focusing on a physical attribute or a specific genetic sequence. Cytochrome B and other mitochondrial genes are commonly used as the separate DNA of mitochondria is much less complex than an organism's full genome, so it's easier to analyze and compare. To identify a monophyletic group, we use synapomorphies, which are shared derived characteristics. That is, a trait or DNA sequence that differs from the ancestral trait, but is shared by two or more groups that also arose from a common evolutionary event. Autapomorphies are derived characteristics that are unique to a specific group. These are not currently useful for finding a group's position on a phylogenetic tree, as they are currently unique. In addition, homologies are traits that are due to a common ancestor, while homoplasies are due to convergent evolution. That is, they evolve separately in multiple species. So we want to find synapomorphies which are homologous. For example, here are 4 organisms, with one short gene shown for each. Normally, these would be much larger, but we'll keep them small for simplicity. So let's arrange them based on shared characteristics, something like this. But we can add more genes to our analysis to increase the accuracy of our tree. Let's add genes 2 and 3 and rearrange our tree. You can repeat this process multiple times by adding more genes, which increases your accuracy, as it weeds out any homoplasies that may have snuck by, and allows you to avoid separating closely related organisms due to autapomorphies present in a single group. And that's how phylogeny works. Simple, right? Let's review. Phylogenies are made of clades, or monophyletic groups, containing a common ancestor and all its descendants. Any individual or species can belong to multiple nested clades, and you can't evolve out of a clade. And to find an organism's position on a tree, we analyze their DNA, or sometimes morphology, especially with fossils, to determine synapomorphies, which are shared traits derived from a common ancestor. And the more traits or genes we include in our analysis, the more accurate our phylogeny will be. I hope this video helped you to better understand phylogenies. And if you have any other questions, ask below in the comments, and I'll answer. If you enjoyed this video, make sure to like, subscribe, share, and maybe check out one of these other videos.